Good morning, friends. Welcome to this screencast. This screencast is totally concentrated on the statistics and the research. What are the various statistical tools that we can apply in research? So, in this screencast, we will discuss about the basics of the statistics that are very important from research point of view. The first one is the population. What is population? It is a complete set of all the members of a group which has specified characteristics like age, gender, geographic locations, income, any disease desired by the researcher for achieving the objectives of research. Means suppose we are going to do a project on some topic like study of perception of students about online courses. So in this the scope of the study is university students. So the target population under this research study is all the students who are studying in the universities. So if I will collect the data from all the population, then it will be impossible for me to contact every individual student who is studying university, because there may be 5 lakh 10 lakh students in a university. So what I will do, I will select some respondent from the university students. Okay, so I will talk it later. So the characteristic of population are known as parameters. So the characteristics of the population, these are known as parameters like mean, standard deviation. These show what are the mean of the responses of the population, how much the mean of population, the responses of the population are deviate from the mean. So these are the parameters of a population. Next is sample. So because it is impossible for me or for any researcher to contact each and every individual who is a part of population and to collect some responses from them. So what is feasible for me, I will select some respondents from the population on basis of some criteria. Okay, so that is known as sample. It is subset of a target population which is expected to truly represent the characteristics of whole population. The characteristics of sample are known as the statistics. So what we do in statistics, by study the sample, we try to estimate the characteristics of the population. We try to estimate about the population. So statistics estimate about the parameters. So in statistics, the whole study is related to the samples, and through those samples we will try to make some estimate about the population. Statistical inferences. The process of inferring something about the population based upon what is measured in sample is called statistical inference. Means when we study the sample and on the basis of the results of the sample, we will try to estimate about the population. This is known as the statistical inferences. Next is statistical estimates. It is concerned with the best estimating a value or range of values for a particular population parameter. So there are two types of statistical estimates, like is point estimate and interval estimates. So what is point estimate? A point estimate is single number that is used to estimate an unknown population parameter. Means when we make some estimation about the population on the basis of just single one number, that is known as the point estimate. Interval estimate, it is similar to a point estimate. What the difference is, in this interval estimate, we don't make estimate on the basis of one point or one number. We make estimate about population on the base of one range. We estimate set of one range. Like in this example, there is a range between 0.17 and 0.77. An interval estimate is defined by two numbers between which a population parameter is said to lie. So that is known as an interval estimate. We set an interval, and according to that interval we estimate about the populations. Estimator, it is a process of selecting an estimate that is known as a estimator, and on the basis of that estimate we will try to estimate the population parameters. So what are the good criteria of good estimator? Which estimator we can say it is a good estimator? One is the unbiased. Means an estimator is said to be unbiased if its expected value is identical with the population parameter being estimated. Means we study the sample, and on the basis of the outcome of the sample we try to estimate about the population. If the results of the estimator are identical with the population parameter estimation, then we can say it is a unbiased. The difference, there is no difference. Efficiency. The estimator with low standard error is more efficient as compared to statistics with high standard error. So what is standard error? We will discuss in later slides. So if the value of the standard error will be high, it means it is more deviate from the mean, so there is a large error. So if the size of standard error is small, we can say the estimator is very efficient. It can estimate the population parameters up to a great extent or nearly equal to 100%. Consistency. So statistics is a consistent estimator of population parameter, as if we increase the sample size, then value of sample statistics must be very close to population parameter. Because while we do any study in management, what we consider as if we will increase the size of a population, the standard error will decrease. So as we will increase the size of a population, the curve will tends to become normal. So we can say when the size of population will increase, the sample size will increase, it gives us a consistent results. Sufficiency. An estimator is said to be sufficient if it makes so much use of information in sample that no other estimator could extract additional information about the population parameter. It means an estimator is said to be sufficient estimator when it gives the maximum information about the population and no other estimator can give more or any additional information as compared to the present estimator. So that estimator is known as the sufficient estimator. Next is standard deviation. It is one of the most important statistical tool that we use in a research. Standard deviation tells how spread out data is. Means when we know what is our target population, and out of that target population we select some samples, and when we try to analyze those samples, then we come to know what is the spread of that sample or the population. It measures of how far each observed value is away from the mean. We try to first we calculate the mean, then we collect the response of a respondent, then we try to find out the difference between the mean value and the response of the respondent. So what is the difference? If the difference is high, we can say standard deviation is high. If the difference between the mean and the observed value is low, we can say the standard deviation is low. It is best suitable tool when the data is normally distributed. When the data is not normally distributed, we can say there is some skewness exists in the data, then interquartile range and semi-interquartile range is the best tool to measure the deviation between the mean and the observed value, or we can say better tool to know about the spread of the data. Standard error of mean that we have already discussed. It is the standard deviation of a population divided by the square root of population size. The standard error of mean tells about the difference between sample mean and the actual population mean, or you can say how much the sample mean is deviate from the actual population mean. If there is a large difference between the sample mean, the responses that we have taken from the sample, and the responses of the expected response mean of a population, If the difference is quite large, we can say standard error of mean is large. And how we can tackle this problem? With increase in size of a sample. With increase in size of sample, the standard error of mean will decrease. So there is some mistake. It is standard error of mean will decrease, not increase. Sorry for that. Next is normal distribution curve. So whenever we collect some data, what is the primary assumption? That the data should be normally distributed, or we can say it must follow a normal distribution curve or bell-shaped curve. So what is the characteristic of normal distributed curve? The curve has a single peak. It is unimodal. The mean lies at the center of a curve. The value of mean, median and mode must be equal and they lie on a simple center of the curve. The two tail of curve extend indefinitely and they don't touch the horizontal axis. These are some characteristics of normal distribution curve. The main characteristic is the value of mean, median and mode that will be same and lie on a center of curve. While we estimate draw a normal curve, there is a one rule, that is 68, 95, 99.7% rule. What is that rule? If we will analyze this normal distribution curve, we will see the data on both sides, left side and the right side, that is equally distributed. In the right side of a diagram that is not colored, if we will observe, in the middle there is a mean. On the right side the values 4.13%, then 13.59%, 2.14%. The similar values are on the left side. What it indicates that the values or the data that equally distributed on both sides, left side as well as the right side. So how we can say there is a relationship between standard deviation and the normal distribution curve? So what we say that approximate 68% of all the values in normal distribution curve lie within plus minus 1 standard deviation from mean. Means the 68% values lies on either plus 1% standard deviation or minus 1% standard deviation. You will say it is 34.13% this side, 34.13% this side. The summation gives us 68.26%. So 68% of all values, the combination of both sides gives always lies within 1 plus minus 1% of standard deviation from mean. Approximate 95.5% of values lies within plus 2 standard deviation of mean. Now if we will come to this picture, right side black and white picture, then we will see the 95.4% values, 34.13 plus 13.59%, it is equal to 47%, and on the left side also 34.13 plus 13.59, again it is 47.6. So combinedly it becomes 95.4% values. So what the normal distribution curve indicates, that the total of 9 values, that is 95.4% values, will lie within plus 2 standard deviation or minus 2 standard deviation. And third one is the total of 99.73% values will lie between plus 3 standard deviation and minus 3 standard deviation, that is summation of 34.13 plus 13.59 plus 2.14. If we will make sum, then it gives us 47. So 49 plus on that side also again 49%. Combinedly it gives us 99.73%, and the 99.73% values under normal distribution curve, that lies within a range of plus 3 standard deviation and minus 3 standard deviation from the mean. This is a very much important concept, and students are not able to understand easily. So it is the best illustration how the values on the normal distribution curve lies. How much area of the normal distribution curve lies within a what standard deviation. Then comes to the confidence level. It is the probability that indicate how much the researcher is confident that interval estimate will include the population parameter. So what is the confidence level? It is the probability that how much a researcher is confident that his interval estimate that he has estimated from a sample will include the population parameter. So if the confidence level is 90%, means the researcher is 90% confident that the interval estimate will include the population parameters. So what is confidence interval? It is the range of the estimate that a researcher makes. In confidence interval, we estimate a range, means plus or negative. We set a tolerance limit. So if a confidence level is 90%, then what we can say, the confidence interval will mean plus 1.64 into standard deviation, that is the upper limit, and mean minus 1.64 into standard deviation, that is a lower limit. Like we set a range equal on negative side as well as positive side. In other words, we can say we set a tolerance level, how much the value will vary on both sides. Next come to the parametric and non-parametric test. This is the foundation of the statistical test. So what are the parametric tests? Under what condition we have to apply those parametric tests? What are non-parametric tests? How they differ from parametric test, and under what condition we have to apply the non-parametric test? So come to the first, what is parametric test? In parametric test, the researchers assume that data are drawn from normal distributed population. So what we assume, that the data that has been drawn from the normal distributed population means the population, our target population, is normally distributed, and the sample that we have selected from the population that are from normal distributed population, and the data follows a normal distribution curve. So for example, the tests like t-test, ANOVA, these are the parametric test. What are the main characteristics of parametric test? The curve is well-shaped curve. The well shaped curve means these tests follow the normal distributed curve, means the data is normally distributed. We assume this main assumption. Mean is equal to median is equal to mean. So it is used to compare the mean between the different groups. If we have to compare the mean between two groups, then we will apply t-test. If we have to compare the mean between three or more than three groups, then we will apply ANOVA test. So we will discuss these tests in another video. Parametric test is used when variables are continuous and measure on interval and ratio scale. This is an important characteristic of parametric test, that it is applied when the variables are continuous and measure on which scale, interval or ratio scales. If the scale is nominal and ordinal, then we will not apply the parametric test. Then non-parametric test. In non-parametric test, the researcher assumes that data are not drawn from the normal distributed population. Means the population, our target population, is not normally distributed and it does not follow normal distributed curve. So we also call it as a distribution free test. Some examples are Mann-Whitney test, Kruskal-Wallis test. These are the examples of non-parametric test. So what are the characteristics? The curve is not a well-shaped curve, there is some skew. Means the data is not equally distributed under the curve. It may be left side skewness or right side skewness. The sample size is normally considered under 30. Outlier exists, means outlier are the extreme values that are totally different from the normal range of the values that are under consideration. Like if the average of all the values 24, then maybe there are some values like 100, 80, or -2, -4. These are known as extreme values that are very much deviate from the mean. We call them as outliers. Non-parametric tests mainly focus on calculating the difference in median, whereas parametric tests, they are interested in calculating the difference in mean. Non-parametric tests, they focus on calculating the difference in median. It can be applied on ordinal and nominal scale. When some ranking is given, we can apply the non-parametric test. So what is the main disadvantage of non-parametric test? If we will compare it with parametric test, how it differs. The interpretation of result of non-parametric test is not easy as compared to parametric test. There is some tough calculations, or their results are not easy to interpret. Non-parametric tests are statistically less powerful when data is approximately normally distributed. So what we can say, if data is not is approximately if the data is approximately normally distributed and we apply the normal non-parametric test, then we can say that these tests does not give the appropriate results. These are the 95% powerful as compared to the parametric test. So it is more advisable to always apply some parametric test as compared to non-parametric test when the data is normally distributed. Now this table shows under what condition which type of test is used. When we have to compare between the two independent groups, compare the mean between two independent groups, if the data is normally distributed, then we will apply independent sample t-test, and its equivalent test in non-parametric under non-parametric is Wilcoxon rank sum test. If the data is not normally distributed, then we will use Wilcoxon rank sum test. Compare mean between same groups under different conditions. Means we have to examine the change in the group before experiment and after experiment. And if the data is normally distributed, we will apply a paired sample t-test. If the data is not normally distributed, then we will apply non-parametric test. And in non-parametric test, the equivalent test is Mann-Whitney U-test. When compare mean between 3 or more independent groups, then we have to measure the one-way ANOVA and parametric test, and under non-parametric test there is Kruskal Wallis test. The fourth is when we have to estimate the degree of association between the two quantitative variables. When we have to analyze or test how much the variables are associated with each other, under parametric test we call it as a Pearson correlation test. And when the data is not on the interval data, it is some ordinal data, some rank is given, then that is a non-parametric data, and we will apply the Spearman correlation test. So this overall table shows that under the condition what are the various equivalent tests on the parametric and non-parametric list. So thank you all. So in this video we have analyzed what is the basics of statistics that is used in research: population, sample, standard deviation, standard error, parametric and non-parametric test. So in next videos we will give detailed explanation of t-test, chi-square, ANOVA, all the tests with the help of some software. Thank you all.